just going to do it, they got a message that regulatory people are going to visit. So they remove the product from the plan and put some simple product so that the area is not very congested. Because these plastic vials, they sometimes fall from the line and show a bad situations. So to regulatory, majority of people, they put a good presentation. So they change the product to some glass vial, 30 ml glass vial. And on that day, that regulatory inspection was over. So next day, they started with alactadine. Now, when the vials were kept back into the quarantine after changing the plan, the worker kept those vials near to the moxifloxacin. Next day, when alactadine was taken, the manufacturing chemist took certain bags. Each bag contains around 1,000 vials, and on the bag there is a big label containing alactadine batch number, expiry date, and that bag is picked up and brought to the labeling line. While labeling, this is a sealed vial, sterile vial, and on the machine it runs and label is pasted and batch number, expiry date, etc. coded. After 3 or 4 hours labeling, it was found that the number of bags which would have been left for alactadine is not that less as should have been. Means they are more than they should be. So what was the meaning? Means something else has come into it on the labeling line. The workers thereafter, they picked moxifloxacin. And when there is a work, those who have worked in factory, we unfortunately have not trained ourselves to read. We don't read. We go by the size, color, location. वहाँ पड़ा है सफेद रंग का, उठा लो उसको. Instead of reading the vial, reading the label, we just what time. First time I may read, second time I don't read. Third time we don't bother at all. Simply go to that location and pick up and bring it. So that happens. So while labeling, along with the olactadine vials, look alike moxifloxacin vials also came to the labeling event. And ultimately what happened? There was mix-up. Mix-up of alactadine labeled vials with moxifloxacin vials labeled, coded with the batch of number of the alactadine. By counting the remaining bag, they could understand. But how to find out which vial? Because the labeling and packing is continuous. As soon as the labels are pasted, then there is a physical inspection of each vial, and then they are put into the shipper after putting into the cartons. So there were some cartons containing both the type of vials. How to find it out? I would like that you think over it. The vial is opaque. The contents cannot be taken off. You open the vial, the lock is broken. That vial cannot be reused. The solution contains viscous substances and is sterile. It is difficult to recover because next time the aqueous solution containing viscosity imparting agent cannot be filtered. And they are mixing the viscous substance and the main solution, they are mixed by aseptic method. So sterility will be lost and cannot be corrected. Vials cannot be reused. And the vial is closed. So there's no mark, nothing. And same 10 ml, 10 ml. If you weigh them, there's no significant difference on the basis of which you can separate the two products. This happened. Now, when it happened, so the manufacturing chemist, QA, QC, everybody gathered in that area. The packaging person that also was there, workers were there, further labeling was taught. The top people, they came to know, they called me, do you know about this problem? I told him, no, I do not know. He told me, please check it, there is some problem. I went there, they told me, this is the problem. So, what are you thinking? So, what they were thinking, how to find out what is the percentage. See, if we take 100 vials, how many vials will be of the other products? This is very wrong thinking. Nobody should have a thinking like this. Even if one vial is going wrong, that is really very serious matter. It is in the category of spurious. And think about the patient who was supposed to take olopatadine, he will get moxifloxacin, or who was supposed to take moxifloxacin, he will take olopatadine. So something clicked in my mind. I had something in my mind that the properties of moxifloxacin is slightly fluorescent. It gives pale yellow color if it is exposed to normal light. But this is a thick-walled vial. Thick means it is around half mm thick. The vial in which is it filled is half mm thick. So I asked the QA, you give me a torchlight. He lit his mobile phone and gave to me. I directed it onto the vials obtained from the control samples of the previous batches, not this batches, of the previous batches. And it was found that the whole of the vial containing moxifloxacin will turn pale yellow from external light itself. And the olopatadine vial did not show any such change. So without destroying even a single vial, the company could separate by examining it against the visual light in the area where MPUs are visualized for visual inspection they are used. So there is always a way to find. I have never handled moxifloxacin, but I do try to update myself with various new APIs, new dosage forms, new products which are coming. So updation is a must. If you keep on doing, you will find you have edge over the others. So for getting a job, you should prepare yourself and be honest in your interviews. Whatever you can do, tell that. What you have done, you can tell that. And if you do not know anything, do not be worried, and nobody will be rejected for this particular purpose. If he fails to tell he, I cannot make, I have not made, I do not know about it, but you are capable of doing it. You find out, explore, develop a method, and do it depending upon or what positions you are. Second part is, once you get into the job, what to do? You see, pharma industry is totally concerned with quality of drugs. All the things, and you should be happy that your job is reserved under the Drugs and Cosmetic Act. No pharma industry can run without having approved manufacturing chemist, and for whom the qualification minimum is only B.Pharm. And for analytical chemist, it is B.Pharm, or it can be a BSc with 3 years experience. So your posts are reserved legally. Now, once you get a job, what you should see so that you can grow that, you can excel others. Please don't be in competition with this idea. He's getting more salary, I should please somebody, people, request him, be in good books, this and that. My experience is you should be good in your own books. You don't have to be good in anybody else's books. Everybody else is watching you. When you go to your company, you are under test. So many eyes are watching you. You are under constant evaluation, even by the worker and by the all persons who will be coming in contact or are connected with your work. So once you join, the first thing is, what is in pharmaceutical? Is good manufacturing practices. People go and attend big seminars, attend conferences, big books. If you are habituated to do your work from your undergraduate level, going through M.Pharm, PhD, or what additional qualification you find out, develop a things, do the things right in the first place itself. And do not use anything which has not been identified or which has not been labeled. If you develop these things, you will develop the concept. These are bases. You will develop the concept, and your hands are slave to your mind. Hands don't work. Hands obey. Feet obey. Only your mind works. So you can carry all knowledge in your head. You read a book. If you have understood that part, it will go to your head. And it will be in subconscious mind and will come out whenever you find its necessity without knowing whether you know it. So when you go to a company, please be clear. You are to develop. It is not I'll do for tomorrow. Those who want to start your career, you must start today. From today onwards, how to do a work. First is how to approach. I ask in interview, when you reach the factory, what do you do the first thing? Then what do you do the second thing? And what do you do the last thing when you come out of the factory? If you have this picture in your mind, it means you have made a plan for working. And if you are totally unmindful, unprepared, unplanned, you will go and then think what I should do today. There is a big gap. You will have to labor to the same extent. But difference in this thinking will make a difference in your whole life. So good manufacturing practices means doing all activities in a planned manner according to the specifications by the authorized persons, by using authorized processes, using authorized documents, using authorized place and machineries, and taking care that deviations are not done. I may claim I know that much. I can make. You tell me any information I can make. Maybe I'm able to make, but I'll not be authorized to do everything, whatever I know. I must stick to the norm. I may be knowing some better methods, but I cannot do it of my own. So for good manufacturing practices, the first part is the commitment of the management. Then there should be quality manual. Then there should be qualified persons. Then there should be written documents. Then all production should be done according to the written documents. Nobody is allowed to make a change, however qualified or experienced that man may be. If any change has to be done, that's called deviation. It should be processed through a deviation protocol, which will be authorized. That change will be generated by the person who will be using or at a user side. That will make the start the protocol, and it will be ultimately authorized by the quality assurance head. Please do not try to copy anybody. Do not try to copy any document, but you may have to copy the format provided by the company or by the organization. If there is none, you can make a format. What are the essentials of the format? Every format must have three things. Every process must have three things. That is time, place, and person. You do anything, your document must demonstrate who has done it, when he has done it, and where he has done it, besides what he has done. Therefore, the good manufacturing practices involves all written instructions, and in a approved format, you can design your own. Sure, the format should reveal all these three things. But when you make, you'll find you have to give places for various things. No unapproved document should be used or be available in the manufacturing area. There is no better instrument than the human eye itself. If you are vigilant, use your eyes and mind together, you may prevent any big mishappening which might have happened due to any unforeseen situations. So by following good manufacturing practices, this topic being very big and required a separate big time to discuss about it, I simply wanted to tell you that good manufacturing practices start from the institute itself. How you handle your practical notebook. I instilled this practice wherever I got opportunity, that in practical notebook you write whatever relevant details in a particular manner every time. And you should write which equipment you have used, how much time you have stirred, how you have filtered, etc., etc. And you will find that GMP is very simple for you. You are already complying with that. Similarly, in say research is thought to be a thing which does not have a railings or limits. It is not so. A research work should be credible only when it has been documented real time. Whether you are doing MPhil or you are doing PhD, please continue. Please apply the same principles of GMP, what you would do after doing your PhD or MPhil. Don't think if that is a different. You see, Dr. Ahuja told me in various fields I have worked, I have never found a difference. People used to ask, earlier you were in production, now you are to R&D. These two things are separate. For me, no, I have not found any difference. Mind is the same. From there, you go to academics, mind is the same. Science is the same. You go to the regulatory, science is the same. People never believed when I said this. But reality is that same principles, same way of working, same concepts are applicable everywhere and they give very good results. So whatever you will build here in the institute, that will become a part of your life. So have good habits, which is called GMP. Once you follow GMP in a factory, you will find that goodness trickles in your own personal life also. You become very conscious and you would like to do everything in your life also with that. Should I answer some questions? Do you want me to answer some questions?
Yes, sir. There are a few questions.
Yes.
So I will let you know. सर आपने एग्जांपल्स बहुत अच्छे बताए और मैं कुछ थोड़ा सा वैसे बताता हूँ जो आपने बताए जीएमपी और ये सब ये स्टूडेंट्स आपने बहुत अच्छा आइडिया दिया। कुछ स्टूडेंट्स का ये है कि यहाँ हम जो रिसर्च करते हैं हमारे पास फैसिलिटी कुछ खास नहीं है, लेकिन जैसे आपने बताया कि हम छोटा सा काम भी कर सकते हैं, एग्जांपल दिए, तो देवर स्टूडेंट्स हु आस्किंग मी फार्माकोलॉजी के यहाँ, जैसे आपने शूटिंग्स के बच्चों को एग्जांपल दिया कि डाइक्लोफेनेक सोडियम पे भी आप काम कर सकते हो, यू कैन मेक दिस फॉर्मुलेशन। सिमिलरली दे आर स्टूडेंट्स ऑफ फार्माकोलॉजी एंड फार्माकोग्नोसी आल्सो एंड दे से कि हमारे यहाँ ज़्यादा फैसिलिटी नहीं है। तो सर कुछ आपके पास ऐसा कुछ एग्जांपल हो, आइडिया हो जिसपे कुछ स्टूडेंट सो दैट they come to know कि भाई हाँ हम इस पे भी कर सकते हैं।
Yes.
Mainly for research students.
Should I speak on that?
हाँ जी सर।
Pharmacology, You see, the most interested part, if you take it by heart, pharmacology, and lot of things are yet to be done, which you can do your own lab, whatever facilities you have got. It's not difficult. You should feel very lucky for pharmacology students that you have animal house. Majority of people, especially in private colleges, animal house is not there. Pharmacology without animal house becomes a big hurdle. In pharmacology, I would say don't go by the big terminologies. For example, this COVID has come. Now everybody will say I'll synthesize drug which will act against this virus. First, I must know what is the virus. I must have a laboratory where I can sustain such activity. And then I should have some concept. I should have a lot of data. But today, the data is lacking, or proper understanding is lacking. So don't run in the dark. My point is, look at whatever laboratories, and I think your laboratory is well versed. I have been sitting with Dr. Dhingra, Dr. Parley. I found on psychopharmacology they have very good. And our one teacher, the most celebrated teacher, Professor S.K. Kulkarni, he used to do all the experiment in his office only. Now this is the selection of design of experiments. What he used to do, he worked on psychopharmacology. He used to take mice is kept in the cage, and they would be near his table and chair also. So he had an instrument, he would drill a hole and instill the drug and put them into the photoactometer and watch their activities. No, all activities in CNS. Such a simple arrangement is required. Such a simple, why students, those who are doing research or MPharm, should not choose such problems where the present space, equipment, and other supporting facilities, they can be done. You see his publications. He was student of Dr. P.C. Pandya Dandia, P.C. Dandia. And he continued his line, and he has published so much of work and students. So the best warrior is he uses whatever weapon is available. Similarly, in research, don't run after the facilities. Whatever facilities, or if there is some point of facility which is must, then you go to some other institute for some time. I again tell you an example. When I was doing MPharm, my senior professor, Dr. K.V. Rangarao was there. He was doing his PhD work, and his work was nearly complete. Only SEM studies was required. Scanning of by electron microscope of his tablets, whether the film coated don't have any defects. The release was through the defects or holes, or what was the mechanism. So in Punjab University, one day he was very sad. What had happened, sir? He was very nice, very gentle person. Nowadays he's in Germany. So he said, my work is almost very good, but it is lacking that electron microscope scanning of my tablets. And I will have to go Bombay, TFRI, and I do not know how much time it will take, maybe 3 months, 2 months, and this and that. So when he told everything, I was surprised. Sir, it is available here. Scanning electron, my SEM, it is available here. He said, what are you saying? It is not here. You see, when I used to study there, I never used to run on the roads. I used to go through the galleries of the departments and reading what is written here. So in geology department, I had read there was a plate where SEM was written. I told him I've seen it. He said, okay, take my cycle. In those days, cycle was the vehicle. So I took his cycle, went there. There, Dr. Kapoor was there. He told me everything, and his problem was solved within 2 days, for which he was not sure how people will cooperate, whether we will be allowed to do something or not. And that professor was so happy. He said, this instrument can be put to use in pharmacy also. So know your surroundings. If something is not here, it is in the biotech department, make a relationship and try to use their facilities. Or say in HAU, please make use of it. Now I come to specific example for cognosy also and for pharmacology also. First, pharmacology. You see, I would strongly suggest that they should try to study screening of non-aqueous solvents required for parenteral injections. Non-aqueous solvents required for parenteral injections. Several drugs are coming which are not soluble in water, so you have to make use of non-aqueous solvents to solve them, or solubilizers. So solubilizers, majority of them are polymeric compounds. Very few. Then there are some good solubilizers, I would like to say. Niacinamide. It's very good solubilizer. But again, how much dose you can give as injection? Benzoate is known very known since ages. Similarly, recently for benefit, benzoates complex with curcumin. Your interest. It has been reported. So for non-solvent, out of non-solvent, you see there are only 5 solvents which can be used in parenterals. And most commonly used is propylene glycol. Propylene glycol has a big problem. It may be contaminated with diethylene glycol or ethylene glycol. Not because they are produced during making it, but because all 3 are same in physical properties and chemical properties. Propylene glycol, ethylene glycol, and diethylene glycol. They look alike. Their physical properties are similar. They are difficult to be analyzed by simple chemical methods. They don't have any double bond in them. So we know they can only be detected with the help of GC. GC is an expensive instrument. So propylene glycol is being used in almost 98% of product where co-solvent is required. And hazardous episodes have taken place, 9 throughout the world. First in 1937 in USA. Very large number of people died. At that time, it was not known whether excipient can show their own action also. And the drug was sulfanilamide. This was the first sulfa antibacterial synthetic drug. For dissolving that, the pharmacist, at that time this word was not there. The chemist, he made a solution in that and administered. So many people died, children and adults both. Thereafter, this testing of API, toxicity of, excipients, that started. Earlier, it was not started because earlier used to be sugar, starch, talc, which were already known to be non-toxic. Then thereafter, this toxic studies of this drug. Present scenario is there are not many, only say 5-hydroxyethanol, phenylethanol, 5-hydroxyphenylethanol. That can also be used. It has a very rosy smell. What you know, it is also used as a preservative. That can also be used as an injection. Benzyl benzoate, it can be used for oily injection, not for aqueous injections. And I told you propylene glycol is most common. And there is one solvent called ethyl lactate. I would request pharmacological MPharm students or PhD students, it's not a big work. One in MPharm it can be taken. Ethyl lactate. It has excellent solubility properties. It is known, it is not a new compound. It is known since ages. And there are certain reports, very small concentration have been used in intravenously in experiments only. It is a green liquid, totally non-toxic. But suitability to the route of administration may have to be studied. It's not available on the literature. It's easily available in India, very cheap, maybe 60 rupees a liter or something like that, equivalent to propylene glycol, this and that. However, it has a drawback. It is an ester. So in alkaline solution, its stability is limited in alkaline solutions. But can be used as in intramuscular injections. No studies. It should be done. Because it have a physical esters, they normally cause burning sensation, even if they are, I do not know this one causes or not. Intravenously, it has no problem at all. But what maximum you see, its hydrolyzed product is ethanol and lactic acid. Both are physiologically acceptable. It is such a good solvent, it can be used in cleaning the machines and put it to the direct drains. In laboratory also, wherever you find some corrosiveness should not be there, you use it as a cleaning agent. It will remove every greasing. It has a very high solubility, non-polar compounds. So they should explore such liquids which have been cleared for their acute and chronic toxicity. Such solvents which are available in the literature, which do not show any chronic or acute toxicity, they should be screened for their route of administration and their concentrations. This is big thing in demand. Why I started with diclofenac, here it is must. I would suggest now, give a project to someone to formulate using ethyl lactate for diclofenac. It's 1 ml ampule requiring 75 milligram of the drug. Already this is there, but they are using polymeric substances which do show anaphylactic shocks. It is not there, this is very little, it's quite frequent in every city, in Hisar, in Delhi, everywhere. It's not particular locality oriented. So similar solvents, finding out, and especially I say, MPharm, such projects should be, which will find industry. Basic principle should be, I should not do a thing which will not find its use. At least in theory, in concept, I should, before starting, I must know if it is found to be non-strong, where it will be used. If it cannot be used, why should I waste my time? It is applied science. It must have applications. Second is, this in pharmacology, don't go for new molecules. Find new uses of the existing. That you can do in your laboratories. Because lot of data is coming for the cardiovascular diseases, and this COVID-19 has changed the scenario totally. It has exposed the possibilities. What are the possibilities of research? Too much, tremendous, starting from your hand spray, going up to your very sophisticated molecules which have come. Here also, I have touched the word hand spray. This is for the pharmaceutics. You should start studying samples from the market which claim hydrogen peroxide on its label. In the market, this hand sanitizers, they are claiming 70% alcohol, ethyl or isopropyl, plus H2O2 plus what else is there? Glycerin. Glycerin is humectant, alcohol is the disinfectant, and hydrogen peroxide is also very quick, very effective disinfectant. But this formula was given by CFDA and WHO as an extempore preparation for the pharmacies in hospital. This is not the formula for the industry. Those who want to give a shelf life of 3 years or 2 years or 1 year. Because hydrogen peroxide is not stable. So we should do a good academic research over it, taking some well-known brands, claiming hydrogen peroxide, putting them in stability chambers, and making protocol. First, make a protocol, how we will do. A committee or 4 or 5 persons, senior person should sit, हमने इसमें क्या-क्या स्टडी करना है, उसको हमने कैसे-कैसे एनालाइज करना है, उसका एक प्रोटोकॉल बना लेने का. Then you give to the students कि आप ये करते रहो, इसका जनरेटर कम से कम 6 months data generation at 40 degrees centigrade, 75% RH. That should be done. And let us see how many products show. I'm sure after 1 year, none of them will show even identification of H2O2.
Some other questions, sir. Let me take very fast. Sir, एक question है. Student ने पूछा है कि when the medicines get जब उनकी shelf life expire हो जाती है, तो what the company do with these expired medicines?
Yes. First, we should understand what is expired. To my dismay, majority people don't understand. I am talking in legal terms. In Drug and Cosmetic Act, under Rule 96, Rule 65, and Rule 109, and Rule 110. Only these are the 5 places where this has been mentioned and defined. Under Rule 109, they have shown how when it is done, 10/22 means 10 is the month and 22 is the year. So the expiry date means last day of the 10th month of that year. Expiry date printed on the drug is a date after which the manufacturer can disown his liability. It is not the drug has actually ceased to be effective. First part is that. I have got trimethoprim sulfamethoxazole got tested after 10 years of its expiry date. Expiry date was 3 years, expiry period was 3 years, and after 10 years, somehow I got a sample during working as a regulatory, and I got it tested. The content was by the analytical technique what we had. Usme wo near to OKI, means within the 90 to 95 to 100% ke limit mein aa gaya. Does that sulfamethoxazole loses activity? No, it doesn't lose. Trimethoprim loses the activity. But the Act says no drug will bear expiry period more than 5 years. That is to give safeguard that even excipient will go under. So while studying this shelf life in the laboratory stability studies, we only analyze the API and its content and any physical changes which are visible. We don't find the chemical changes happening within the API, although we know it is very rare it will happen, but it does happen in certain situations. For example, morphine with amino groups, say lactose, morphine with lactose, the content of morphine goes down. Any sugar you add, content goes down. But the question is, the expiry date has been crossed. Then what we do with this? There is only one way, that you crush it and destroy. They cannot be used to recover the chemical, even if it is stable. It is not perfect because we do not know what other changes in science is not perfect. It is continuous. The data available is not perfect. It is only as we know today. Day after tomorrow, many things may come up. Therefore, the drug which has expired should not be used. It should be destroyed. If you have some dealer is there, and then he should inform the regulatory or may not. After expiry, anybody can destroy. And then the question is how to destroy it. How to destroy it. If these are tablets, you peel the strips and put into water. And capsules like that. Ampules, crush them. Vials, crush them. And there are machines, hammer, Hatak machine, you can crush them. And bury it in a pit. It is not to be thrown into the. Recently, only yesterday, I got a call from the regulatory. There's some news that at somewhere some drugs were found, expired drugs are heaped. So it will cause environmental pollution. Okay, so what should they do? Nothing to be done. Simply you make them unusable by peeling it or by crushing it and bury it in a pit. Nothing is to be done. It is never to be processed, reprocessed. There have been certain reports from Ghaziabad or somewhere else that on one upon a time, some gang was detected. Those who were unpeeling and selling to somebody so that he can restrip the drug. This is unfortunate part and should never be thought about it. This is a proper thing. It should not be done. Yeah. So, and now, any other question?
Yes, sir. The same student has asked, is there any eco-friendly method to destroy the drugs?
Eco-friendly, you see. Let us understand what is eco. In eco, what we say, number one, the gathering of people, those who are living in a particular colony gathering. This is one ecosystem. Second is the water bodies. And third is the land itself. Fourth is the air. If we burn it, this is the best one. Incineration, but it costs money. That is the best part, if we can burn it. And where there are huge quantities, they have to do it. And you don't have to have that incinerator in your university or in your house. You can engage with somebody, but we have to be tracking them because they many times don't burn it, they throw here. Private agencies are there, they are authorized by the government. So the burning is the best way because everything is converted into carbon dioxide, water, sulfur dioxide, nitrogen oxide, etc., etc., etc. Second is you make a slurry of it and put into a pit. Or if there is a big body, big rivers, and your volume is not big, then with the permission of pollution control, they make certain sewerages and use it. But it is highly controlled by laws, so nobody should scatter it here and there. If you put a heap of the drugs, some children, some nefarious persons, they may come and resell it or rebottle it, etc., etc. So it is the duty of the manufacturer, dealer, and everybody handling it that it is not subjected to unauthorized reuse because these are very technical things and people will not understand. They are not supposed to understand. They should be safeguarded with the help of our practices and whatever details are given on the bottles. These are.
Sir, one more question is there. एक बच्चे ने पूछा है कि we were earlier importing API from China, high dependent. Now India has imposed ban on importing API from China. How much this will boost Indian API sector? एक तो ये. And secondly, क्या ये पूछ रहा है, do you think that we will be able to sustain this ban for import from API from China?
Yeah. First, I should say you should feel happy that it is a great opportunity. Those who want to go into this API manufacture, this is the, I don't think you will get ever better chance like the present. It is very golden opportunity. Unfortunately, India has been lacking. Even at NIPER also this was discussed. They were very much interested, but the thing is, the investors, those who want to open factories, they want कि आज से मैं बनाऊं और कल ये पैसे में convert हो जाए. In dosage form, it is easy because they know they have even purchased very heavy, costly material. It is closed in the vials or in the bottles in the same form or in some converted form, and it goes to the market. In API, first is investment is high, but government is supporting it. For the students, I want to say they should go for smaller, simpler things. I'll give an example. Somebody asked me what I can start. You know, your doctor in his heart, psychiatrist hospital is there near that house colony. So he wanted his some relative wanted at Sonipat. So I told him, you go simple things, one step method. If you have low investment, go for one step method. What is the one step method? I gave him an example of ferrous fumarate. You have to have fumaric acid. You don't have to synthesize. You have to have ferrous sulfate. Even if you want to make ferrous sulfate, it's very easy. Simply you make it solution, filter it, and stoichiometrically you have to mix precipitation. You have to filter and dry it. So such simple step, industry is there. Then another is we go slightly bit. I don't say this is the only one. There are in. Similarly, if you go bit more investment, if you can do, go for povidone. These are technically simple. You don't need to buy technology from anywhere. You can improve upon whatever you have your knowledge. My class fellow who is in Jammu, he started with the API. He did B.Pharm, and today he has gone into the formulations also. So API opportunity is very good currently, and a lot of things are available, but their ready-made processes are not available. The unfortunate part is on internet you won't find. But if you have a background and if you want to develop, better is maximum 3 steps for a medium scale. If you want to go beyond that, then you must hire a technology and consultant to start with that. But once you have started, the beauty in API manufacture is that the same reaction vessels are used. Number 2, the reaction vessels don't have to be clean area. They can be under sheds. Only penultimate step. From there, the GMP compliance is required. Final step, one step before the final step. That should be protected from the environment. There, the water, air, personal hygiene, and whatever you want, like that. That has to be complied with. So the basic major structure does not require the cost, which is required in formulation. So this is a very nice area, and so many people are working for it. Similarly,
सर एक क्वेश्चन है बच्चे का, वो पूछते हैं कि कुछ is there any other they want you to suggest some preservatives for eye drops except benzalkonium chloride.
Good. Benzalkonium chloride is a culprit basically. Some people have used it in injection also. This is a quaternary ammonium compound. It gives pain at the site of injection if it is used. And in Nathan Life Sciences, I got it changed. And when we contacted the person, because that company is contract manufacturer, they were making for some company, we told them, they came and discussed. They say, yes, we are getting complaint on injection. Our injection gives pain. Simply, I told them, you have to do this. Put benzyl alcohol. For eye, very good preservatives, phenyl, 5-hydroxyphenyl ethanol. 5-hydroxyphenyl ethanol, yes. ये common है, liquid है, इसकी solubility limited है, but it is used for eye, and it will give a very good flavor of rose. In the rose, the fragrance what we get is because of this 5-hydroxyphenyl ethanol. It's cheap.
Okay.
1.5% to 2% you can use in items. It's very compatible being an alcohol. It is not phenol, it is alcohol. So it's very, very compatible to ice and very stable unless there is some instability due to hydroxyl group, any particular drug. Otherwise, it's very much one should go for this. Yes.
So there is one more question. One student has asked. Uh, she says that some companies used to practice to sell their product. They change their category into OTC and cosmetic. Is there any monitoring regulatory?
Yeah, I know. You see, regulatory system in India is very poorly structured in terms of manpower. You see in Hisar how big this area is. In Hisar city itself, at least near to 1,000 shops are there when we total the wholesale and retail sale. And there is only one drug inspector and one senior drug inspector, right? So this OTC and first we should understand cosmetic. There's no word called Ayurvedic cosmetic in the drug and cosmetic. No license given to any Ayurvedic under the name of Ayurvedic. It can contain Ayurvedic ingredient, means plant-based ingredient or earth-based ingredient or other than APIs. Excipients which are other than APIs, they can be used in making cosmetics. And OTC drug, then it is a drug. So OTC drug means over the counter, it is again not included. OTC word is not included in drugs and cosmetics. There's a Schedule K in the drugs and cosmetics. They have given a list of substances which can be purchased without medical practitioner. And I'll tell you, for the knowledge of all students, if they are taught to this forensic, they are taught forensic pharmacy, the form course. So they should go through the Schedule K. They must go through the Schedule K. And second thing is key, for example, glucose bottle. Where does it fall? Glucose bottle. Can I purchase a glucose bottle directly from a chemist? Yes. It is not a scheduled drug. And then if we think why it is not a scheduled drug, we will understand why certain drugs are on prescription, why certain drugs are not on prescription. It is not that except for the Schedule K drugs, no other drug can be purchased without prescription. Any drug which does not fall under Schedule H, H1, X, or Schedule G, or NRX, means narcotic drugs. One rest of them can be purchased without prescription on and without supervision of pharmacist. But no license will be given unless he has engaged pharmacist. Suppose I have to sell amoxicillin, pharmacist supervision is required. I have to sell any drug, even cotton, on prescription, then pharmacist is required. But a man does not bring a prescription, wants a cotton, one can sell. The idea is when doctor writes, it means it is specifically for some treatment. And the pharmacist or seller cannot know what will be the implication there. So that must be checked by pharmacist before selling. And any drug where it is in Schedule H, it means it contains such substances. I would say you see certain vaccines, no Schedule H requirement. Because they don't contain a substance which, if taken, not supervised by medical profession, can cause any harm. Anything which can cause harm until unless it is supervised medical professional, they can be sold without prescription. So all the 548 drugs in Schedule H, it means if they are taken for unwarranted conditions, the person may face certain bad situations, adverse effects. So doctor has to choose that although this drug has this adverse effect, but his disease is like that he will choose to give it despite of the adverse effect. So scheduled drugs, they have to be on prescription. Now we say what is OTC? OTC, that is, as the student is understanding, I'll talk in his language. OTC, what he understands, over the counter drug. This term is used in other countries, not here. But it does mean that these are not necessarily governed by the prescription. So they are being sold as a cosmetic. There are certain errors found by the licensing authorities also. Recently, they issued a license under cosmetic for this hand sanitizer, which is a drug. So they should have got, now those have been cancelled. To 30th June, they have been cancelled. So the marketing people always try to bypass the restriction imposed on them. They devise language, ways, and putting, can you believe, in one of the eye drops, which was a product of a very big company in India, which deals only in eye drops, they were adding vitamin B12. And they were having competitive edge. And that B12 as excipient, they are not giving, it contains it. And excipients, glycerol, this, and B12. I put my foot down. They said, this is a big O, and they are making since years. So they came there, he tried to, we will tell to the court, sir, nobody is going to ask you why you did it. They will try you, you say whatever, you will go under its poorest category. ये explanation यहीं इस रूम में आप बोल सकते हैं, वहाँ कहीं रिकॉर्ड में नहीं आने जो आप करें, ना आपको मौका मिलेगा, आपकी जस्ट कहाँ है, कहीं नहीं है, that you can use as excipient as bitter. So marketing people to get a marketing edge or interesting one. बीटामिथासोन क्रीम सबसे ज्यादा किसकी बिकती है? ग्लैक्सोस की बेटनोवेट. So betonate used to come in green color carton. Betonate. So trade name is betonate. It is mandatory to write betamethasone valerate cream IP. So generic name also. In IP, this cream was not official, and they were claiming IP. I was very much confounded. Why nobody else is making? Only this is they are making. Then I checked the IP, BP. In BP, both are official, ointment as well as cream, betamethasone valerate. In IP, only ointment was official at that time, and USP both were official. I issued notice to Lexus. I was ready to prosecute. My department did not give permission, but I had given copy to the Drug Controller India. They directed the Maharashtra authorities. They had to stop. Now it comes in blue and with proper name. Now they make BP, betamethasone valerate, BP. So the bigger companies, they try to take advantage of their being big. These are marketing people, and they always study how we can say this our product is the product, ऐसी कोई product नहीं है. तो regulatory का काम भी यही है. The common man is not supposed to learn this and go to details. So OTC versus cosmetics. The simple thing is, you see, its license. If it starts with cosmetic COS, it is a cosmetic license. If it is manufactured by MFG LIC, it is a manufacturing license, means for the drug. If he is claiming any API contains ye itne percent. For example, Borolin. Borolin understood to be cosmetic. No, it is not cosmetic. It is drug. And there has been a prosecution with this regard, because boric acid and zinc oxide, they are drug, they have therapeutic value. So definition, whether it is a drug or not, depends upon the intended use. If it is to be used for the treatment, prevention, diagnosis, or mitigation of any disease or disorder in human beings or animals, then this is a drug. And if it is used for beautifying and cleaning the human body, not for veterinary, for human body, then it is a cosmetic. So the marketing people, they try to project this is a cosmetic, everybody can. You see, Vico Vajradanti advertisement, no? They repeat this word. This is a drug. It is not a cosmetic, right? To make a decision, or somebody can sue them. So it's there, but majority cases it is not. Yeah.
तो सर क्वेश्चंस तो दे आर जो पूछे थे रेलेवेंट क्वेश्चन हमने आपसे पूछ लिए एंड इसका कोई एंड नहीं है वैसे बच्चों ने जो पूछे थे रेलेवेंट क्वेश्चन आए वास्ट यू बट बड़ी इंफॉर्मेटिव थी सर आपने बहुत अच्छे एग्जांपल्स दिए सो और भी फ्यूचर में कुछ होंगे तो हम आपसे और इंटरेक्शन करेंगे और
you see had it been a gathering then things are more understandable, no? It is through online. Yes. You see, I'm not able to see their reactions, no facial reactions. Anyhow, we have to be used for this method, and it has so many advantages. You see, nobody has to go out from his place. So, Dr. Manish, you are doing really very wonderful job. Yes. I'm really happy, and I thank Madam Niru Vasudeva for.
I would. आपने जो एग्जांपल दिए हैं बहुत अच्छे रहे हमारे बच्चों को उससे काफ़ी हेल्प मिलेगी फॉर देयर रिसर्च वर्क आल्सो सो आपने बड़े सिंपल आइडिया आपने इतने सिंपल ढंग से बताया तो बातें जो हैं हमने भी काफ़ी सीखा है मेरे कोलीग्स ने भी सीखा होगा मैं रिक्वेस्ट करता हूँ मेरे कोलीग मेरे जो को कोऑर्डिनेटर हैं डॉक्टर विक्रमजीत सिंह टू प्लीज कम लाइव एंड एक्सप्रेस हिज वोट ऑफ थैंक्स
Thank you, sir. So, on behalf of Department of Pharmaceutical Sciences, I am highly thankful to Shri Rajendra Kharna ji, sir. He has been so kind to spare time for us, and he has given very valuable instructions and tips to the students so that they can enhance their skills in research as well as their academics. He has given very small ideas which they can use to pursue their research work, and we hope he will be there to help them as and when required, and all the faculty members are also thankful to him for his suggestions from time to time. I am thankful to the participants as well as they have been very patient in listening this talk, and I am thankful to Chairperson Niru ma'am and Professor Manish Ahuja sir, because he is the one who is regularly taking pains to arrange these different seminars and conferences and these online webinars for the students, which will benefit them in their career progression. Thank you, sir. Thank you very much, sir.
Thank you. So nice.
Thank you, sir. Thank you. We are highly grateful to you, sir. So, dear participants, now we finish our talk here. Thank you for coming here. Thank you, sir. So, I am ending the session. Thanks.